Hello everyone. I would like to start another exciting and interesting lesson today. Before that, I would like to start by asking some question. Have you ever heard of a disease which produces this type of lesions? The case fatality of that disease may even reach to 100% in untreated cases. More than 1 billion people are at risk of this disease, which is very alarming. Or have you ever heard of a disease in some states of India causes acute encephalitis syndrome, especially children under 15 years of old? The case fatality is as high as 75%. What about this one? Have you seen this type of lesions? The case fatality of this disease ranges from 10 to 90%. So who is the culprit for this transmission of these deadly diseases from human to human or animal to human population? Yes, this lecture is about that culprit. I'm welcoming you all to another interesting video lecture on medical and veterinary entomology. In this lecture, I'm going to discuss on a very important vector of different diseases, that is sandfly or phlebotomine. First, I will introduce the zoological classification and important genera of different sandflies. Then I will highlight the morphological features, life cycle, bionomics, medical and veterinary significance of sandfly. Finally, I will conclude this lecture by highlighting the prevention and control measures of sandfly or phlebotomine. Follow my channel's playlist to get more video lecture on parasitology. Stay connected with the channel to get more video lecture on different subjects of veterinary medicine. So, let's get started. This is the zoological classification of sandflies. Flies under the family Psychodidae are called psychodids. They are small nematoceran flies having long antennae, pendulous palps, long legs, and hairy bodies. Under the Psychodidae family, there are two subfamilies, namely Phlebotomini and Psychodini. Flies under the Phlebotomini family are commonly known as phlebotomine or sandflies. In this lecture, I will talk about sandfly or phlebotomine. The subfamily Phlebotomini comprises more than 800 species classified into 6 genera. The New World genera are Lutzomya, Brumtomya, and Oroya. The Old World genera are Phlebotomus, Sargentomya, and Chinius. About half of the species are contained in the genus Lutzomya, 1/3 in Sargentomya, and the majority of the remainder in the genus Phlebotomus. A small number of species remain under the genera Brumtomya, Oroya, and Chinius. Overall, around 464 species are considered as New World species and 375 species are considered as Old World species as of today. The genera or subfamilies are distributed globally but absent from most oceanic islands and Oceania, including New Zealand. Remember that the New World genus Lutzomya and Old World genus Phlebotomus included species of public health and veterinary importance. What are the important species of sandflies? Keep watching until the end. General morphological features of sandfly or phlebotomine. Adult sandflies are brownish, long-legged, narrow body about 3 to 5 mm in length. Their bodies and wings are hairy, so you can say almost full body is hairy. Mouth part are short to moderate in length. Mandible is well developed in female, therefore female are hematophagous. Mandible is absent in male. We know that in most of the arthropods, male bear pilose type of filamentous antenna and female bear plumose type antenna, but it is different in case of sandflies. Both male and female sandflies bear pilose type antenna, and each antenna bear 16 segments, which is often have a beaded appearance and thickly covered with hairs. Using the pilose type antennae, male cannot recognize sound of female like mosquitoes. Pulpa recurved, hairy, 5-segmented, third segments bears sensilla. Based on the pulp, New World and Old World species can be differentiated. But how? Note that the third pulpal segment is longest in New World species and fifth pulpal segment is longest in Old World species. Wing also helps in identification of sandflies. Wings are lanceolate shaped, hairy as mentioned earlier. It is held erected above and roof-like over the abdomen during resting conditions. The radial vein 2 and 3 and radial vein 4 form fork, which is located at the middle of the wing. Remember that in mothflies, forks are located on the inner side of the wing. There are 4 larval insters in the life cycle of a sandfly. Diagnostic features of the larval instar included the presence of 1 or 2 pairs of long caudal setae. The first instar larva possesses 1 pair of caudal setae, and second to fourth instar larva possesses 2 pairs of caudal setae. The mature larva, that is fourth instar larva, is grayish-white with a dark head. Antennae are small and leaf-like. Head bears chewing mouth part. The abdominal segments of the larvae bear ventral pseudopores, which is unjointed evagination from the body used for progression. So, based on these pseudopores, thorax and abdominal segments can be differentiated. Pupa is exarate type, that is, legs and wings are free from the body and can be seen externally. This type of pupa is also known as true pupa. Pupa possesses short prothoracic respiratory horns. General life cycle of a sandfly. The life cycle of a sandfly comprises 3 developmental stages such as adult, larva, and the pupa. On average, a female sandfly deposits 30 to 70 eggs in protected places such as in dark, cool, humid niches or any moist places. For an example, Phlebotomus papataci deposits 40 to 80 eggs at a time when the temperature is above 15 degrees centigrade. So, these eggs hatches between 4 to 20 days after oviposition. Remember, prolonged hatching time is required in cooler weather or any unfavorable conditions. There are 4 larval instars in the life cycle of a sandfly, as mentioned earlier. Depending on temperature, availability of food, sandfly species, stages of larval instars, larval development is usually completed within 20 to 30 days. Note that larvae feed on organic decaying material, leaf mold, insect bodies, rabbit feces, etc. The larval developmental period may prolong to up to 7 months if diapause occurs, especially for the larval instar 4. After that, pupation usually requires around 2 weeks. Remember that pupal stage is very sensitive to desiccation. After the pupation period, adult flies emerges during hours of darkness. Life expectancy of adult sandfly is not clear. Male may survive only for a week, whereas female may live longer in comparison to male fly. Female sandfly feeds multiple times before developing viable eggs for oviposition. Note that autogeny, that is ability to produce eggs without requiring a reproductive male, is also evident for sandflies. Bionomics of sandfly or phlebotomine. Old world phlebotomines are savanna and desert species, whereas New World phlebotomines are mainly inhabitants of forest where high rainfall prevails. Most of the Old World or New World species are exophilic, that is, they tend to inhabit or rest in outdoors. A few species are endophilic, arthrophilic, and zoophilic. You need to know the definition of terminologies that I mentioned. Endophilic means these species remain closely associated with human and their domestic environment. Arthrophilic, those species which feed on human. Zoophilic species are those species which prefer to feed on animal and man. Depending on different environmental factors such as temperature, humidity, host availability, and habitats or breeding places, a species can be adapted as exophilic or endophilic or zoophilic. I have listed some of the species here as example. Just have a look at it. Most of the sandfly species are exophasic. That is, they feed outside of dwellings. Only female are hematophagous, but sandfly feed on plant sap, nectar, honeydew, and blood of wide range of animals from domestic to wild animals including humans. Feeding time of sandflies also vary depending on different environmental factors, host availability, but most of the species are nocturnal and crepuscular. That is, they feed during the time of twilight to different time of night. Some of the species can feed the host during day at shaded condition. What are the breeding places of sandfly or phlebotomine? Adult sandfly are found in and around the location where suitable resting places are available. Breeding or resting places include dark, cool, humid niches like cracks and crevices of soil, rocks, manure, tree trunks, tree hollows, nests, tree couches, animal burrows such as the burrows of rodents, stables, livestock pens, buildings, forest, litter, caves, well, or any moist protected places. Sandfly or phlebotomine is important as pest and vector of different diseases of man and animal. Sandfly are very annoying biting pests. They bite repeatedly and cause a sharp prickling sensation each time. Biting of some species may cause sleep difficulty in highly infested areas. A study demonstrated that the mean bite ranges 20 to 50 per person per night, even reach up to 300 bites per night. The biting lesion included pink or red papule about 2 to 3 mm in diameter and 0.5 mm high. Moderate to severe itching usually occurs. The lesion remains prominent for 4 to 5 days and then start to disappear. Individuals who are hypersensitive may develop hives with pronounced swelling at the biting site, such as eyelids and lips. Sandfly act as the vector of leishmaniasis. So what is leishmaniasis? Leishmaniasis is a protozoan disease caused by more than 20 species of leishmania. There are two major forms of the disease such as visceral leishmaniasis or Kala-Jar and cutaneous leishmaniasis. Mucocutaneous leishmaniasis is another form of the disease. The most common form is cutaneous leishmaniasis. Visceral leishmaniasis is the most severe form of the disease. Mucocutaneous leishmaniasis is the most disabling form of the disease. Remember, people become rigs not due to the bites of the sandflies, but due to the leishmania transmitted by the sandfly. Visceral leishmaniasis is characterized by irregular intermittent fever, substantial weight loss, swelling of the spleen and liver, and serious anemia. If not treated, the fatality rate can be as high as 100% within 2 years of infection. Cutaneous leishmaniasis produces ulcer on the exposed part of the body, such as on the face, arms, and legs. This lesion may even lead to disability. Mucocutaneous leishmaniasis produces lesions that partially or totally destroy the mucous membrane of the nose, mouth, throat, and surrounding tissues. PKDL or post-Kala-Jar dermal leishmaniasis, a complication of visceral leishmaniasis, it is seen in apparently recovered patients. PKDL is seen in most countries, but it is mainly seen in East Africa and Southeast Asia. You can see global distribution of leishmaniasis. Globally, more than 1 billion people live in endemic areas who are at a great risk of this infection. An estimated 3,000 new cases of visceral leishmaniasis and more than 1 million new cases of cutaneous leishmaniasis occur annually. Sandfly act as the primary vector of the Chandipura virus. I would like to give you some brief information about this disease. The Chandipura virus is a bullet-shaped virus like rabies virus under the Rhabdoviridae family. It was first detected in 1965 in 2 humans in Maharashtra, India. It is thought an occasional pathogen of human, but in 2003 an outbreak occurred in Andhra Pradesh state of India and case fatality was 56%. In 2004, outbreak occurred in Gujarat state and case fatality was 78%. According to the World Health Organization, the recent outbreak occurred between June and August 2024, which is considered the largest outbreak in the past 20 years. Overall, the disease is known to cause sporadic cases. An outbreak of acute encephalitis syndrome occurs in western, central, and southern part of India, especially during monsoon season. In Gujarat state, outbreaks typically occurs every 4 to 5 years. The case fatality ratio of Chandipura virus infection is high, ranging from 55 to 75%. The disease affects mostly children under 15 years of old. Symptoms include high fever, diarrhea, vomition, convulsion, coma, and some cases deaths. It can lead to high mortality within 2 to 3 days with the onset of symptoms presenting with acute encephalitis syndrome. Primates and in some countries, hedgehogs act as the reservoir of this infection. Note that different species of Phlebotomus and Sargentomia species have been reported as major carriers of Chandipura virus. Other than sandflies, mosquitoes and ticks can act as vector of this deadly virus. Sandfly act as the vector of Bartenollosis or Carrion's disease. Carrion's disease is a disease of human caused by a gram-negative bacteria called Bartonella bacilliformis. It is a biphasic illness. The acute febrile anemia is called Oroya fever, which is a serious life-threatening phase of the disease, particularly for the children. The case fatality ranges from 10 to 90%, which is very alarming. In chronic or benign dermal eruption form of the disease occurs in previously exposed inhabitants called Peruvian warts or Verruga peruana. It is characterized by pain in muscle, bone, joints, and finally appearance of warts as skin lesions. The disease is mostly distributed in Andean valleys of Peru and in some areas of Ecuador and southern Colombia of South America. Sandflies act as the vector of Changuinola fever virus. This virus primarily infects a wide range of birds and mammals. In human, the disease is characterized by febrile illness. The virus is mostly distributed in Central and South America, occur widely in Panama, Colombia, and Brazil. Other than sandflies, this virus has also been isolated from mosquitoes, sloths, armadillos, and rodents such as rice rats act as the reservoir of this infection. Sandflies act as the vector of sandfly fever virus or flavivirus. It causes sandfly fever or Papataci fever, which is a non-lethal, self-limiting illness of usually 3 days duration. The disease is characterized by fever, headache, nausea, limb and back pain. Encephalitis may also occur in infection with Naples serotype of this virus. Rodents and primates act as the reservoir of this infection. Sandfly act as the vector of vesicular stomatitis virus. This virus has also been detected from different flies such as black flies, biting mites, houseflies. It is particularly important for livestock, which causes acute febrile, weakening disease of horses, cattle, swine, and also sheep and goats. In this viral infection, blister can be seen in mouth, on feet, teeth, and also other parts of the body. It is also known as pseudo foot and mouth disease due to its similarity with foot and mouth disease. So in summary, Sandfly act as the vector of leishmaniasis, Chandipura virus disease, Bartenollosis, Changuinola virus disease, Sandfly fever, and vesicular stomatitis. Prevention and control of sandfly or phlebotomy. Sandfly bites on the exposed areas of the body. Repellent lotion or spray such as DEET, which is very effective and useful. One can use on exposed skin when going outside. People are trying many alternatives of repellents such as any citrus juice, eucalyptus oil, coconut oil, treaty oil, tiger balm, etc. I am not sure about its efficacy and duration of protection. Wearing protective clothing such as long sleeves, trousers, and socks should be worn in visiting sandfly infested areas. Remember that sandfly cannot bite through the outdoor clothing due to their short mouth parts. When sandfly are present, the campsite, outdoor sleeping areas should be dry and open to the wind, and obviously should be away from the breeding places or sites. Interestingly, sandfly can even pass through the standard 16 to 18 mesh mosquito netting. So what about the alternatives? Will you use fine mesh mosquito net? No, it will impair the circulation of the air. Better to use insecticide treated standard mosquito net, wide mesh net jackets and hoods, head nest, screens, tent openings, bed nets may provide good protection. Treated bed net for sleeping indoors and treated window screen door, and bed nets for indoor protection. Synthetic pyrethroids insecticide including pyrethrin, cypermethrin, etc. are recommended by the World Health Organization for the treatment of bed nets. Indoor spray of natural or synthetic pyrethroids insecticides provide excellent protection in indoors during the daily period of sandfly activity. Insecticidal smoke by mosquito coils also useful, and popular control measures for indoor spaces. Organochloride, organophosphate, carbamide, and synthetic pyrethroid insecticides are effective for residual control of adult sandflies. In many countries, treatment of housing, including walls, ceilings, windows, etc., are treated with these chemicals mentioned. Environmental measures for sandfly control include elimination of breeding and resting places, filling cracks and crevices of wall, ceilings, floors of houses, outbuildings, cleaning outdoor areas, relocating the piles of manure in case of farmhouses may reduce the prevalence of sandflies. Biological and genetic methods for sandfly control have not been demonstrated, but scientists are still trying for the best one. Thanks for watching, and if you have any question, please write it in the comment box. I'll be happy to answer. And good luck for your studies. I'll catch you in the next lesson.